That's good. Hi, my name is Anna Rakowski, and I am the Cure CMD chairman. And I have with me Dr. Anna Ferrero from Paris, France, who is here to talk with us about her plans to launch a clinical trial in the SEPN1 related myopathies. So first, I just wanted to ask Anna to tell us a little bit about herself.
Well, actually, I'm a neurologist. I'm a practicing neurologist and a molecular biologist also, and I run a lab which focuses on studying early onset myopathies, that is, inherited muscle conditions, which involve children. And we look at the genetic basis of them, at the mechanisms involved, and we look for therapies that we can translate into treatment for these diseases.
Great. Would you want to share with us a story maybe about one of your patients that helps kind of describe what a SEPN1 related myopathy patient might look like?
Well, a typical SEPN1 related myopathy patient is pretty normal at birth. Some of them are floppy, but only in the neck and trunk muscles. And the most typical thing is that they are unable to properly hold their head at the age of 2-3 months when they are supposed to start having head control. Because again, the neck and trunk muscles are much weaker than the other muscles. Limbs are good, so they generally start walking at a normal age, but they remain unable to lift their head when they are lying on their back, and they need to turn around and push on their arms to get up from their bed. But they're usually doing pretty well till school age, and by that time usually there's a drop in their weight, they lose fatty tissue, they become very skinny, and most of them develop scoliosis, that's a back deformation, and also a respiratory failure that needs medical assistance.
Great. What I'd like to talk about now is your plans for a clinical trial in the SEPN1 related myopathies, and maybe you could share with us initially how that came about, what led you to make a breakthrough discovery that allowed you to identify a treatment to take forward to clinical trial.
Well, actually, we wanted to understand what is the function of the protein that is lacking in these patients. And for this I had the contribution of Sandrine Arbogast, who is a specialist in oxidative stress, because on clinical and molecular data we suspected that oxidative stress was involved in this disease. So we took muscle samples from the patients without cellular protein N, we put them in culture, we grew these cells, and we looked at what happened in them. And actually we saw that in the lab these cells had markers indicated that they are producing more reactive oxygen species, that is, they have chronic oxidative stress. And we could see that when we treat these cells in the lab with certain antioxidants, and particularly with one which is called N-acetylcysteine, these cells recover the normal state, they extinguish, they tone down all the markers of oxidative stress, and they recover survival and resistance that is pretty close to normal anatomical cells.
Wow. When are you planning on launching the clinical trial, and how do you see that moving forward?
Well, actually, the first thing we need to check is whether this drug is as effective in animals and in humans as it is in cells in the lab. So we're going to do in parallel studies in the mouse model of cellular protein N deficiency, and also in a selected and small group of patients to see whether the drug is well tolerated, which should be, because this drug has been on the market for 40 years and it's very safe. And we're particularly going to look at how these muscles, which show oxidative stress markers, react, and whether these markers are corrected after 6 months of treatment in the patients in the trial, in the pilot trial, and after 6 weeks of treatment in the mice. And I think this would be a good indication of effectiveness in humans of this treatment, and that will be an important step that will allow us to move forward and start a clinical trial, double-blind placebo, with several reference centers at the international level.